Okay, so thank you guys for having me. This is a really special symposium for over the 3 days. I also am really impressed by the Saturday morning science series. I think there should be more of this outreach between scientists and the local community. So I think this is a really special thing you guys have here. Happy Pi Day as well. This is the only actual Pi Day of this century, 2015. We missed 9/26, but soon we'll get to the Pacific Pi Day time. Okay, so I'll tell you guys a little bit about the work we do in lab. I'm going to try to sort of sweep through a century or two of studies of inheritance over the first half of the talk. As we get into the second half of the talk, I'll get more into our research. And so what we're interested in is the topic of epigenetics, which is what this whole symposium is. Epigenetics is sort of a fraught word because for many reasons. One of them is that there are many different definitions that people use for the word epigenetics. I'm going to distinguish between epigenetic inheritance and epigenetic marks, okay? So I'll define my term here. By epigenetic inheritance, what we refer to is a trait that's heritable from one generation to the next, either cellular generation or organismal generation, which is inherited in the absence of underlying changes in DNA sequence. And so the easiest way to describe this is that basically every cell in your body has the same genome, right? You all came from one fertilized egg. But when a liver cell divides, it never makes, where's the laser weapon? When a liver cell divides, it never makes a skin cell or a kidney cell, even though the skin cell and kidney cell have the exact same book of instructions. It makes 2 liver cells. So genetic inheritance is the copying of DNA. This is the copying of the book of instructions, whereas state liverness is an epigenetic state, okay? And this is like a set of bookmarks. So this is epigenetic inheritance. Studies over the last couple decades in lots of different model organisms, for example, fruit fly, have identified a lot of the machinery underlying cell state inheritance. And so here's the typical kind of thing you can find when you Google epigenetics. 2 of the major carriers of epigenetic information in terms of the molecular carriers of epigenetic information are histones. Okay, so 6 feet of DNA in each of your cells is wrapped up into a 5 to 10 micron nucleus. It's compacted 100,000-fold. The way that happens is you sort of wrap the DNA, about 150 bases of DNA around an octamer of 8 proteins. So these things can be modified in a wild number of ways. There's something like 100 different chemical groups that can be added to the histones. We also have, you think of 4 bases in the DNA, A, G, C, T. There's the so-called 5th base, which is methylated cytosine. Methylated cytosines can also be copied during replication. This image doesn't show some of the other famous epigenetic information carriers like small RNAs, which we'll get into shortly. Now, it is the case that in some paradigms, histone modifications are passed on from one cell to the next. However, not all histone modifications are passed on. So this is where I'm going to distinguish between epigenetic inheritance and epigenetic marks. All of the stuff that happens attached to the DNA you can call epigenetic marks. Epigenetic inheritance refers to when these marks are being passed on. Okay, so this is part of the reason that this is a complicated word, is that sometimes people say epigenetic and they mean marks, and sometimes they mean inheritance. So today I'll be talking about epigenetic inheritance. Now, this is basically the last time I'll talk about mitotic epigenetic inheritance when cells pass on information. It is worth pointing out that defects in epigenetic information underlie lots of interesting and important diseases. So there's an increasing awareness that epigenetic misregulation plays a role in cancer. And I think one of our speakers tomorrow will be talking about this. But mitotic epigenetic inheritance, cell state inheritance, when 2 cells divide and pass on information, is an interesting and important field. And this is sort of our day job. In yeast, we work on how you pass on histones to 2 daughter cells. So one analogy I like to make is if copying your DNA is like photocopying a piece of paper, copying your chromatin is like photocopying a piece of origami. And mechanistically, this is a really interesting question. So that's sort of our day job. However, for multicellular organisms, not yeast, but flies or humans or corn or something, We don't divide like this, right? So we don't pass on a lot of juice from one generation to the next. We go through the choke point of gametes, the sperm and the eggs. So for information to be passed on from one multicellular generation to the next, it has to survive the process of gametogenesis, making the sperm, making the egg, and then at the sort of moment of fertilization, you make a zygote, and then in order to affect the offspring that develops from that zygote, the child, that information has to sort of unravel in a developmentally complex and interesting way. So I'm going to be talking about this, what people call intergenerational or transgenerational epigenetics, and we're sort of interested in if this ever happens, when it happens, why it matters. Okay, so this in the end is the study of inheritance. And so just to orient everyone, to understand inheritance, we have to understand the basics. So this all goes back to Mendel and his peas, which Tracy mentioned recently. So Mendel studied 7 traits in a little pea patch, whether or not the pods were inflated or deflated, white or purple flowers, smooth or wrinkly peas, yellow or green. And what he found was that if you crossed, let's just pay attention to the color here, a true breeding, so plants can often inbreed, they can have sex with themselves. So he had true breeding lines of yellow and green peas. So he mates a yellow with a green, and he gets all the kids are yellow. Then he lets these guys self with themselves, and he found this reproducible across many different traits, 3-to-1 ratio of offspring. So basically, out of all of these, you would see 3 yellow peas among this guy's progeny and 1 green. Okay, and this gave him, he introduced the concept then of dominant and recessive alleles. So the terminology you'll often see is, so many organisms are diploid, which means they have 2 copies of each chromosome, right? We're diploid, we get a copy of our genome from mom and a copy from dad. So if you consider the trait for yellow peas as a big A and for green peas as a little a dominant allele is the big A. So if you have a big A and a little a, you get the big A phenotype, which here is yellow. Now, if you cross these guys, your sperm and your eggs, your pollen and ovules, you'll get a big A and a little a out of this guy and a big A and a little a out of this guy. And only when you get 2 little a's together do you reveal the recessive trait. Okay? So this is called a Mendelian inheritance pattern. And the reason this matters is because epigenetic paradigms often involve non-Mendelian inheritance. So I'll give you guys a couple examples of pretty classic epigenetic information paradigms. These often show up in plants for reasons that we can get to. So one of the classic examples is the phenomenon of paramutation, which was discovered by Brinks and Co. in the 50s, again with plants, with corn. And so basically they found that you have a difference in color between plants. They can either be purple or green. Now, when you mate the purple with the green, you get green offspring. Okay, so here you would think that the green would be the dominant allele, and the purple would be the recessive. So maybe this guy is big A, this guy is little a, and now you should have a big A, little a, right? So this would be what would happen in Mendelian genetics. If you allow this corn to cross with itself, it turns out that it just keeps making green corn. You don't get 1/4 of offspring turning purple. And in fact, if you mate this green guy with another purple guy, so if you had a big A, little a, and you crossed it with a, little a, you'd expect half your kids to be purple, but all of them are green. And so what happens is that this piece of DNA can actually convert this piece of DNA to its state, okay? And this is not DNA editing. This is a change, as it turns out, in all of these epigenetic marks, okay? So actually cytosine methylation, histones, and small RNAs. Just to orient you for a moment, notice that there are 7 little arrows. These are 7 repeats of the same piece of DNA, and we're going to come back to this concept. All right, so 7 identical copies of, I think it's a 200 base pair stretch. This turns out to be really important for paramutation to work. So this is a plant version, and again, as I mentioned, plants do lots of non-Mendelian inheritance. This also occurs in mammals, so one of the best characterized cases is something called the Agouti variable yellow mouse. And you can see, again, we have a coat color. A lot of this stuff was discovered because of really easy to track phenotypes like coat color. So here we have a gene which is responsible for the color of a mouse's coat. The AVY mouse, it turns out, has a little copy of a selfish element called a transposon. We're going to get to these. They were discovered in Sanborn Field. It has a little transposon inserted near the promoter of the gene. And so genetically identical mice can have a range of coat colors from yellow to brown. Now, brown mice have kids who skew towards brown, and yellow mice have kids who skew towards yellow. Okay, so again, this is non-Mendelian inheritance. And again, the molecular mechanism underlying this involves things like cytosine methylation, that fifth base. This is a case of sort of random variation in an epigenetic trait. People call it Epivariation. There are also programmed cases of epigenetics, and a medically important one, which is a useful and instructive example, is in the domain of imprinting diseases. So imprinting diseases are diseases where a piece of DNA seems to know what sex it came from in the parent. Remember, we have 2 copies of everything. So here are 2 different syndromes. They're called Prader-Willi syndrome and Angelman syndrome. They're quite different phenotypically, okay? So Prader-Willi children are obese and hypogonadal. They're both developmentally delayed. I forget, people used to say mentally retarded, and I don't know what you're supposed to say now. But both of them are that. So Prader-Willi kids are obese and hypogonadal. Angelman syndrome is very characteristic. They're atactic. They do a lot of inappropriate tongue thrusting and smiling, and they often were referred to in the early days as happy puppets. And so these are quite different from each other. It turns out both of them come from the exact same lesion to the DNA, okay? So in principle, you could have 2 human beings with the exact same genome, okay? This is a deletion of a part of the chromosome called 15q11 to 13. Each of these people could have a deletion of 1 copy of 15q11 to 13 and be massively different from each other in terms of their phenotype, okay? And it turns out the difference between them is whether or not that deleted piece of DNA came from mom or came from dad, all right? And so in this case, this piece of DNA remembers which sex it came from. It remembers whether or not it came from the egg or it came from the sperm, all right? So this piece of DNA exhibits some memory. Now, in terms of why this gives you these different phenotypes, this is a little cartoon, well, sort of cartoon, cartoon for scientists, of the 15q11 locus. And what you see is that there are a bunch of genes which you have 2 copies of, but these genes are only expressed from 1 copy or the other, from your maternal copy, the copy that came from mom, or from your paternal copy. And so what happens is normally you express this copy from mom, but you're not expressing dad's version, or express this copy from dad, but you're not expressing mom's version. So now if you delete this locus, what happens is, even though you have both of these genes, so normally when you delete 1 copy of a gene, it doesn't matter. Most genes can function in 1 copy. 1 copy is enough. But here what's happening is that you're only expressing the maternal version of this. This gene, you would only normally be expressing the paternal version. So by losing dad's copy, you're not expressing any of this. So this is like having a double deletion, even though it's only a single deletion. Okay, so this is why, and because it's different genes that are absent when you're missing the dad's copy or you're missing the mom's copy, this is why you get these wildly different outcomes of this deletion. Okay, so imprinting, paramutation, and epivariation are 3 of quite a few different transgenerational epigenetic inheritance paradigms. And as with cell state inheritance, the mechanisms underlying them include all the same players. Okay, so I showed you 1 cartoon of this, here's another cartoon. So you basically have histone modifications, you have DNA methylation, and now we have teeny RNAs. All of this gets very complicated to study because all of these things talk to each other. So cytosine methylation gets directed to the DNA by small RNAs. Cytosine methylation affects the expression of small, blah, blah, blah. These are all sort of interconnected, and so it's often challenging to understand exactly what is being passed on in the gamete. But in any case, these are the molecular mechanisms for many epigenetic inheritance paradigms. So, one of the other things I have to tell you before we get into the thick of it is that when you eliminate these things, so if you make a mutation in a histone modifying gene, or if you make a mutation in DNA methylation, it turns out that one of the most common things that happens, what's the point of all this machinery? Why do we have all this stuff? It's not to have a range of coat colors. It turns out that all of these things play a role in dealing with selfish elements in our DNA. Okay, so you guys have probably heard that the genome has 50% or 90% junk DNA. What people often mean by that is transposons. Okay, so as I mentioned, these were discovered by Barbara McClintock in the 1940s. She eventually won a Nobel Prize for it, like 40 years later, here in Sandboard Field. And she was studying basically corn kernel coloration in maize. And basically what she discovered is that there are these little hopping DNA or mobile DNA elements, which are called transposons. And so what you have is if a transposon is getting in the way of expression of a pigment gene, then you will be one color. If it jumps out, then you will start getting the pigment gene expressed and you'll get the other color. And so part of the way she infers this is by patterns of sectoring in these kernels. So here you're not getting expression of the purple, and that's what happens in this case. Here, this gene jumped out, the transposable element jumped out early before this kernel started developing, and so you get the whole kernel one color, and then you get these splotchy patterns where as the kernel is growing, the thing jumps out, when the kernel is like 10% this size, and then the cell where it jumped out gives rise to a little patch of color, okay? So it turns out that depending on your genome, I think with humans we're something like 50% of these selfish elements. So when they hop out, they can also copy themselves. So we have thousands of copies of transposon type 1, hundreds of copies of transposon type 2. Okay, so we have tons of these things in our genome. And it turns out that when you eliminate all of this sort of molecular machinery of epigenetics, the main thing it does is transposons stop being silenced. Okay, so all of this machinery, histone modifications, cytosine methylation, small RNAs, presumably first originated as a way of shutting off selfish elements in our genome, but then because of where selfish elements are, this can often lead to sort of other kinds of phenotype. So for example, now we're going to come back to epivariation in the Agouti mouse, where you have the yellow through brown. The reason that mouse shows that is because a transposable element happened to hop near a coat color gene. Okay? And so that's why you end up getting this phenotype that isn't really about transposons, because a transposon is near a gene for something else. All right? So what I'd like to leave you with in terms of that concept is that epigenetic machinery is devoted to control of selfish elements, and because selfish elements are all over our genome, that has all sorts of other consequences for gene regulation and for our phenotypes. Now, selfish elements are not all bad. In fact, if I eliminated all the selfish elements from one of your genomes, it would be completely sterile, because it turns out that we've also harnessed selfish elements to do useful things for us. And the example I'm going to come to at the very end is that we use selfish elements in control of the placenta, okay? So the placenta is an invasive process, right? The baby's taking goodies from mom. One of the ways this happens is that cells in the placenta fuse with each other. They form what's called a syncytium. The protein responsible for that is called syncytin. Syncytin turns out to be a viral surface protein called envelope, which we have harnessed to use to make placentas. Okay, so in the sort of evolution of mammals, this has actually happened at least 6 different times, where different placental mammals have picked up a different retroviral ONV protein to allow the formation of these syncytial cells. Okay, so this is called exaptation when we use someone else for our own purposes. So we use selfish elements in control of a lot of processes that we need. So selfish elements are not only selfish, we make use of them as well. So all of this is basically preamble. I'll give you guys one more thing, which is that many of the epigenetic marks, histone modifications, et cetera, listen to the environment. So DNA doesn't listen to the environment. If I eat a hamburger, it's not going to change my genome. But if I eat a hamburger, there will be histone modification changes in my chromatin. So this is, again, my day job with yeast. This is mapping of 26 different histone modifications. This paper is coming out in 2 weeks. And we put yeast into a stressful environment. And here, just looking at one gene, you can see that the levels of different marks, this guy comes up. This is something you see on active genes. This guy comes down. So histone modifications and many of these other things change in different environmental conditions. Okay? So we know that some epigenetic information makes it through our gametes into the next generation. We know that some epigenetic information listens to the environment. And so this has resurrected an old idea, which is that the environment in one generation might be able to influence the phenotype in the next generation, right? Depending on which of these things make it into sperm. So the inheritance of acquired characters is often called Lamarckian inheritance, after the early evolutionary theorist Jean-Baptiste Lamarck. He's the subject of a lot of ridicule and derision, which I think is unfair because, as I understand it, he was one of the first people to understand that evolution needed a theory, that species changed over time. So he's famous for the idea of the giraffe's neck. So giraffes would sort of reach to get to leaves higher up on a tree. It would get a longer neck. It would pass on the longer neck to kids. And so over multiple generations, you would have longer and longer necked giraffes. Maybe a little bit of a less crazy version of this is the blacksmith. So he would be pounding iron all day. He'd have big arms. Maybe his kids would have stronger arms. So here's a sort of a comparison of Darwinian and Lamarckian evolution. So with Lamarck, the giraffes sort of reach up to the leaves. They get longer necks, and then over multiple generations, you have giraffes with these long necks. The Darwinian view is that you have variability in the neck length in a population. This guy who can't reach these leaves dies because he can't eat. And then these guys breed with each other, and so as a result, you have longer necked offspring. So there are two things I want to show you with this slide. One of them is that Lamarck understood, if you think about this idea for about 2 minutes, you'll understand that we don't pass on everything that happens to us to our children, right? So soldiers from wars throughout history who've lost their arms don't have kids without arms, okay? So Lamarck had this sort of goofball idea that there was an element of desire or need. So if the giraffe wanted those leaves, then he would pass on the longer neck, okay? The other thing I want to show you about this slide is that it sort of illustrates a pitfall in giving talks. So when you give talks a lot, you sort of get into a rut where you're sort of doing the same thing. Tracy's already heard this story, which is sort of meta. So for a couple of years, I would do this thing where I was showing the slide where I'd say, I apologize for the poor quality of the picture here. It's surprisingly hard to find a picture of a dead giraffe online. You know, I thought you'd like Google dead giraffe and there'd be like 50,000 hits and you'd be like, oh, weird, like enthusiast clubs. So anyway, I always did this, right? And so I was giving this talk at the American Society for Andrology like 2 years ago. And so after the talk, I'm talking to a couple people, and this guy comes up to me and he goes, I can get you a picture of a dead giraffe. I was like, no, dude, that's like an internet joke. But so he's a really nice guy. He's a Brazilian andrologist and a big game hunter. So he talks to us for a couple minutes. He gives me his card. And he has to go meet someone for lunch, so he leaves. And I say, bye, thanks for the card. Email me that picture when you get home. They're like, oh, I don't have that picture yet. Did I just kill a giraffe? Okay, so you have to be careful what you say. Okay, so the classic test of the inheritance of acquired, and I should also mention, both Darwin and Lamarck believed in the inheritance of acquired characters, so calling this Lamarckian inheritance is a misnomer. Like A, both Darwin and Lamarck believed in the inheritance of acquired characters, and B, this is by no means the goofiest part of Lamarck's theory, the inheritance of acquired characters. But in any case, it has gained the term Lamarckian for whatever reason. So the classic test of this was carried out by August Weismann. He's the person who introduced the distinction between the soma, the cells of our body, and the germline. Okay, the germline is your sperm and eggs and the stuff that turns into them. And so what he did is to test the inheritance of acquired characters is he cut the tails off of mice, he mated them to each other, and he measured tail length in offspring. He saw no difference, and so he concluded that you do not inherit acquired characters. Now, there are at least two problems with this experiment. One of them is the more you learn about the thinking at the time, it's a little bit of a rigged experiment. Again, amputees don't have children without arms, and even in Lamarck's version of this, you had to have this element of desire. Okay, so to some extent, you wouldn't have expected, even at the time, tail length to change. And the other thing is that if your parents told you that someone was cutting their tails off, maybe the useful thing to do would not be to have shorter tails, but it would be to run away from people in lab coats. So he may be measuring the wrong thing. So in any case, this has been sort of, this was one of the nails in the coffin, so to speak, of the inheritance of acquired characters. The discovery of the structure of DNA was another. But in the last decade or two, a number of people, many of whom, well, some of whom are in the audience, or just spoke, have been revisiting this type of paradigm where you poke an animal and you look at the kids. So now I'll tell you guys about our system. So what we do is we do this as a paternal effect paradigm. So no doubt moms pass on lots of information in the oocyte, but mom is also baby's first environment. Okay, so fetal alcohol syndrome doesn't have to be magic and epigenetic, you're poisoning a fetus, okay? So whereas dads, if you do it right, just leave sperm in a memory. So if you do it right from our point of view. So what we do is we split male mice to brothers to two different diets, control and low protein. The reason we choose low protein diet is that the types of things it makes sense to tell your kids about should be things that are mildly toxic, that change on the order of every 10 to 1,000 generations. So we sort of thought that this access to food might be one of those things. We raise mice from weaning at 3 weeks of age to sexual maturity. We mate them with females for a day or two, then we take them out of the cage, so they're not influencing their children through cultural inheritance or something like this. We're trying not to let them influence their kids through the microbiome, although they probably do defecate in the cage at least once. We then let the females be pregnant, have kids, then we grind up the kids, and we look at gene expression by microarray. So there are basically two important distinctions between this experiment and the Weissman experiment. One is we've chosen an environment that it might make sense to tell your kids about. The other is that instead of measuring one quantitative character, which is tail length, we're measuring 30,000 or 30,000 genes in mammals. Okay, so when we do this, and we're looking at the liver of the children. It turns out that hundreds of genes change depending on what your dad ate. So I don't know how many of you guys have seen microarray clusters, but basically each of these columns is a comparison between two livers. Red means that a gene was higher in a low protein liver. Green means it was higher in a control liver, or lower in the low protein liver. Whoa. And so what you can see basically by seeing a big block of color, that means for the majority of offspring of this cross, 80 to 90%, a subset of genes were turned on. Okay, so about 400, 500 genes can tell what your dad ate, even though all dad is contributing here is basically an ejaculate and a day of memory. Okay? Now, one thing I'd just like to point out, so here we're looking at the absolute levels of one of these genes, and so you can see the low protein offspring sort of shift up in each case. These red triangles are higher than these. But you see quite a bit of variance, and one thing I'd like to point out, people have very deterministic ideas about epigenetics sometimes. Epigenetic here, the paternal diet is maybe contributing to 5 to 10% of the variance in the expression of this gene. There's a bunch of other stuff that happens between control animals. Okay, so epigenetics is like a quantitative character. So complex diseases like diabetes have many different inputs. Epigenetics is one of them, but it certainly doesn't mean that it's going to force you to be a way that you really don't want to be. It'll just make you more likely to gain a little bit of weight on some diet. Okay, so it's not, epigenetics is a quantitative contributor to this kind of stuff, but it is not by any means deterministic, I would say. So what are the genes that are changing? It turns out that the genes that are upregulated in low protein offspring's livers are basically include lots of cholesterol and lipid biosynthesis genes. So this is the pathway for the synthesis of cholesterol. Each of these boxes is a gene that plays a role in this pathway, and they're colored by how they change in low protein livers, again the kids. And so you can see they're all red. So you coherently upregulate the cholesterol biosynthesis pathway if your dad ate a low-protein diet. Is this just a sort of gene expression phenotype, or is there something else going on? Is there a metabolic phenotype? So we look at lots of metabolites in the kids, and we see a very dramatic decrease in cholesterol esters and a subtle but significant decrease in free cholesterol. So what we think is happening is that the offspring of low-protein dads are losing cholesterol and cholesterol esters somehow. Cholesterol esters play a role in bile acids, so maybe they're dumping a lot of bile acid into the gut. And then they probably turn on cholesterol biosynthesis to keep up with this loss. Okay? So this is our paradigm. We're not really interested in deep metabolic phenotyping, so this is all I'm going to say about metabolism, but there are a bunch of other things that came out around the same time that have similar conclusions. So here's just a couple of the papers that have come out in roughly the same time period. This is the paper that pushed ours out the door. So here in rats, males on high-fat diet fathered daughters with altered glucose control. Okay. This is basically our paradigm. It's a low-protein diet, and they also see things like blood pressure changing, and I believe they see glucose control here. These are both cases where you're taking a male from weaning to sexual maturity. Many more studies do things where they'll starve a pregnant mom. So all of these studies from Ferguson Smith and Patty are undernutrition. These from Tracy Bale are for overnutrition. So if you give an altered diet to a pregnant mom, you can then take her son as the paternal treated generation and look at the next generation. So these are paternal effect paradigms, but where you're messing with dad while he's still in the uterus versus a child. Okay? And all of these link ancestral food supply to metabolism in future generations. Now, it turns out that we certainly didn't mean to do this, but this models human epidemiological studies. So the most famous of these is the so-called Dutch hunger winter. So at the end of World War II, in response to a transit workers' strike, the Nazis imposed an embargo on food shipments to the Western Netherlands. 20,000 people starved to death. Government rations were on the order of 700 calories a day. People were eating tulip bulbs to survive. So there were pregnant women in the country at the time, as there are going to be. And it turns out that children who were starved during the Dutch hunger winter have significantly elevated rates of diabetes, obesity, and cardiovascular disease. So this has led to something called the thrifty phenotype hypothesis. This basically holds that if your parents tell you you're going to go hungry, it makes sense to hoard every calorie you can get your hands on. When conditions of plenty return, this can become maladaptive. Okay? It's actually interesting. People have proposed that this has something to do with the fact that first-generation Americans, whose backgrounds are from third world countries, tend to have very high rates of diabetes. That may or may not be accurate. It's difficult to disentangle from socioeconomic factors like the McDonald's effect. Now, this is the maternal effect paradigm, and as I told you, maternal effects are sort of hard to separate from, mom has uterus and lactation and everything else. This also shows up as a male line paradigm. Tracy mentioned this before, the so-called Uvercalyx cohort. This is a remote parish in Sweden where they started keeping really good crop records in the 19th century. And it turns out that modern epidemiologists have linked basically disease rates and mortality rates to your ancestral food supply. And what you can see is that your risk of diabetes, obesity, and cardiovascular disease in a grandson is linked to the nutritional status of his paternal grandfather. Okay, so a male line effect in humans on metabolism in future generations. So all of these things together link ancestral food supply to metabolism in the future. Since I think I'm going on a little bit longer per slide than I'd like to, I will not mention many of the other types of things you pass on. One of my favorites is this odor paper that came out last year from Ressler and Diaz. But we can get to other things that are passed on besides metabolic status if anyone wants to in the questions. So we're mechanists at heart. So the question for us is, how does this work? Again, all of this stuff has to go into the gametes and come out. So this is a charming slide that my French postdoc made. I like it for multiple reasons, including the implication that you're grinding up Mickey and Minnie babies. But the point is, of course, everyone's assumption here is going to be that this information is in sperm, right? Many other things do get passed on from dad to children besides the sperm epigenome. So first of all, in humans, there's the possibility, because we're outbred, there's the possibility of selection on sperm. So maybe in Uvercalyx, the guys who didn't have enough food, all of the sperm that had A at position 500 died, and all of the sperm with G at position 500 survived. Now, that's mostly not a problem in animal models, because we use inbred mice. So they don't have different copies of anything anywhere. So we don't think this is a sort of genetic selection, but we can't rule out that there's a programmed change in the genome. Okay, so that's one. Two, it turns out that mom can influence children through cultural inheritance. So this is the review. These are studies by Meany and Schiff. So rats differ in the extent to which they care for their children. This is licking, grooming, and arched back nursing. And so a mom who cares for her children will have kids who grow up to be caring moms, and moms who don't care for their children have the same thing. So here you have mitotic epigenetic inheritance, where the kids will remember as they grow up how much they were cared for. But the information transfer between generations is actually cultural. It's how much mom is licking the kids. Okay, so this is again mom. It turns out that moms can judge dads and change how they treat kids accordingly. So my favorite example of this is the Gouldian finches. These are birds. They come in red, black, and I guess yellow. The experiment's done with red and black. So what these guys did is they took, if you made a red male with a red female or a black male with a black female, you get a large number of large eggs that are both sexes. If you made a red female with a black male or vice versa, you get a small number of small eggs that are mostly male. So who cares? They're genetically different. It turns out that what they did is if you take a red male and paint his head black, a red female will give him a small number of small eggs that are mostly male. Okay? So the only information being transferred is photons. The ecological thinking about it is that if mom doesn't get to choose a mate among many, and she has a suboptimal mate presented to her, she will mate with him, but she'll save herself for her future kids. Okay? This is where all the guys are shaking their heads grimly. Now, in principle, in our system, moms could be judging the low-protein males as inadequate and restricting blood flow to the placenta. Okay? Finally, it's worth pointing out that there's a lot more that goes into copulation than sperm. So we also generate what's called seminal fluid, which is juice from a bunch of different glands along the reproductive tract. And literature largely in flies shows that seminal fluid can alter female post-copulatory feeding, mating, and other kinds of things. Okay? There's also some literature in mammals, largely from pigs, like prostaglandins are in the seminal fluid. So it could be the case that the low-protein males have altered seminal fluid components. Okay? So this is sort of now the way we have to think about this. You put males under different conditions. We have different dietary ones. So we mostly do low protein. We also look at high fat. We look at nicotine now. Tracy looks at stress. We mate them to females. We see alterations in the kids. So you can imagine that it's in the sperm epigenome, but you could also be altering seminal fluid or cryptic maternal effects. So experimentally, how do we separate these? You could do it by artificial insemination, but that's actually very challenging in mice. But what we can do is in vitro fertilization. Okay? So we just take sperm and eggs and we mix them in a dish and we make the kids. So now our lab, we do all mouse breeding through in vitro fertilization. They don't get to have sex anymore. It was all they had. So we do IVF. Here's the first clutch of embryos we made. We're really proud of this because I still think of us as a yeast lab. And so this was our first set of litters. We now have something like 50 litters analyzed. And so sure enough, we find that the same genes, these are all cholesterol metabolism genes, are upregulated even when you use low-protein sperm in vitro fertilization. So we know that sperm carry at least some of the information, or it co-purifies with sperm. Okay, so all of that seminal fluid stuff was a red herring. So in the last little bit here, I'll just talk about our favorite lead for how this works. One thing I'll point out about IVF is that IVF, it's a pain to learn how to do, but once you do it, it has a lot of unanticipated advantages. When we do IVF, because you only need a little bit of sperm to make embryos, in principle you need one sperm to make an embryo, What we do is we take like 5% of a sperm sample from males, we fertilize a clutch of embryos, and so then we can implant them, and this is where we look at metabolism in the kids, but we also have access to the early embryo. We can also make embryonic stem cells out of them. But what's really interesting is that we can archive 95% of the sperm sample, and so we can take 2 sets of offspring who have different cholesterol metabolism, and we can retrospectively look at the epigenetic marks in the very same sample of sperm that was used to make them. And this turns out to be really, really powerful for both epivariation, control to control variation, and basically really powerful for anything where we have low penetrance. Okay? So we can look at how many differences there are in epigenetic marks in sperm and figure out what fraction of those differences got maintained during fertilization and early embryogenesis. So this is we really like this pipeline. So we've been, even before we knew the information is in sperm, of course, we've been characterizing the sperm epigenome in great detail. We have leads in all 3 of these domains: histone modifications, cytosine methylation, and RNAs. I will tell you for 3 or 4 slides about our favorite lead in RNAs. Okay? So small RNAs are famously involved in lots of really well-characterized epigenetic inheritance paradigms. Paramutation uses small RNAs. RNA interference in worms, which won my colleague Craig Mello the Nobel Prize, is a small RNA-mediated phenotype. So small RNAs are sort of involved in lots of these transgenerational things. So we sequence small RNAs from sperm, isolated from control or low-protein males. So what's unusual about mammalian sperm, this is the length distribution of small RNAs. These are up to 40 nucleotides. Unlike most somatic cells, the vast majority of small RNAs in sperm actually come from tRNAs. tRNAs are the RNAs that link amino acids making a protein to the mRNA. So these are the things that transfer information from the mRNA to proteins. So sperm clip all these things in half. And so like 70% of small RNAs in sperm are what are called TERFs, tRNA fragments. They come from the 5' ends of tRNAs. And so it turns out that several of these TERFs change levels dramatically in response to low-protein diet. Now, this is very intriguing to us for a bunch of reasons. One of them is that tRNAs directly see amino acids. We're using a low-protein diet, so there's sort of a very obvious way that you would sense low-protein diet using these things. They're very abundant, and sperm are very small and have very little RNA in them. So how would you tell that's the only thing you're really giving to the oocyte. And then the really cool thing is that, and I didn't know this, and I bet even the professionals here don't know this, tRNAs are universally used in retrovirus and retroelement replication. So this is HIV. In the virion, HIV genome is packaged with a tRNA bound to what's called the primer binding site. When HIV infects a cell, this tRNA primes reverse transcriptase. That's the thing that makes the DNA copy of the HIV genome. So you make the complementary DNA to one so-called long terminal repeat. You come over to the other one, you make the whole genome here. So because of that, and this is universally conserved, all retroelements, all retroviruses, even though there's this arms race between hosts and pathogens. So because of this, by clipping your tRNAs, you could fight off any retroelement you might see. So basically, any retroelement I could get from another human being, I could fight, even if my genome's never seen it, I could fight off by cutting my own tRNAs. Now, and all the more reason not to procreate with non-humans. As I mentioned, because epigenetics is about control of selfish elements first that gets co-opted for other things, this actually got us very excited about these RNAs. So we don't know that these are responsible. We're doing experiments now where we're injecting them into zygotes. But everywhere we look, they're neat. Okay, so on the biogenesis side, it turns out that we don't think that these tRNA fragments are made in the testis during spermatogenesis. We think that they're actually made in this long convoluted tube where sperm mature after they're finished developing, called the epididymis. So here you can see tRNAs and tRNA cleavage. They're not cleaved in the testis, they're cleaved in the epididymis. As sperm go through the epididymis, they fuse with these little vesicles, which are like fake viruses, that would be called exosomes in other contexts. They're called epididymisomes here. Epididymisomes have the exact same RNA payload as mature sperm. And in vitro, we can take immature sperm from the caput, fuse them with kata epididymosomes, and we can show that we can deliver these small RNAs to sperm. So this is basically our hypothesis at this point, is that the testis is where sperm learn where all their transposons are, and the epididymis is where sperm learn what's been going on in the world around the male. Okay? This is very compelling for us for a bunch of reasons. One of them is that small RNA production in gametes increasingly turns out to have an element of soma to germline communication. So this happens in fly oocytes, in ciliates, in plants. My favorite example is pollen here. So a pollen grain has 2 sperm nuclei, which are the germline, and a vegetative nucleus. The vegetative nucleus releases all transposons from repression. They destroy its genome. It's littered with breaks from the transposons jumping around. So it commits suicide. By doing so, all the transposons are expressed, and they get clipped into little antiviral RNAs and shipped over to the germline. Okay? So there's this possibility that the soma is where you make dangerous RNAs to ship to the germline. So one thing that that's really compelling for us is that we may have found the mammalian spermatogenesis version of this. So that's biogenesis. In terms of function, when we interfere with this tRNA fragment in embryonic stem cells, we de-repress about 30 to 40 genes. Okay? We've seen these genes change in the early embryo when we use control versus low protein sperm to make the embryos. Everything smells right. Again, we haven't done the injections into zygotes yet. But it turns out that every single one of these genes is regulated by the long terminal repeat of an endogenous retroelement. So we have a tRNA fragment regulating an endogenous retroelement. All of these genes are involved in the placenta. Okay? And so all of the metabolic phenotypes we see in kids, and everyone sees in paternal effect paradigms, also show up when you mess with the placenta. So our working model is that by changing the level of tRNA fragments in sperm, dad regulates an exapted endogenous retroelement, which plays a role in placentation. You change the size or the function of the placenta, and you get these metabolic consequences as a downstream effect of altered placentation. So that's where we are. This is sort of the cartoon of everything. It's totally insane, but at least half of it is right. With that, I will tell you the people who did the work in my lab. So this is my lab as of like 5 years ago. The mouse team has largely been Jeremy Shea, Lucas Fouquier, and Ben Caron in the early days. Marcus is doing the nicotine project. The main people behind the work on the small RNAs in sperm are Upasana Sharma, a new postdoc. And I actually don't have Colin Conine, but he was our collaborator in Craig Mello's lab. Here are many of our collaborators, and thank you for your attention.
Thanks so much, Ali. We're going to take questions for about 8 or 9 minutes and then go straight into the next talk. So I'm not sure where Massimo is, but you might want to come up and get your stuff set up. Questions? Someone? So, okay, here we go.
So have you made the cross mixtures of the epididymosomes from fed and unfed and vice versa, and you get what you expect?
Yeah, so that's a really good idea. We're not sure, so these RNAs do change in epididymosomes, but not as much. And so one of the things we find is that different small RNAs are gained at different parts of the epididymis. And so it may not be the case that you're regulating the levels of epididymosomes, levels of RNAs in the epididymosomes, but you're actually altering how long sperm take to get through the epididymis, in which case doing that fusion experiment with control or low protein epididymosomes won't recapitulate things. But this is so the big thing about mammalian reproduction, why not do this kind of stuff in flies, worms, or plants where you have better genetics, is that I think uniquely in mammals you can mechanically disassemble and reassemble reproduction. So you can take early sperm, give them vesicles, put them into oocytes, So anyway, we're doing a lot of these kinds of things now, but a negative result wouldn't invalidate the hypothesis.
I have a couple of questions. First, did science just get lucky that Mendel chose peas instead of, say, corn because of the effects of epigenetics?
Yeah, well, there may have been, many Mendels before then who were studying things like human height, right? Human height is non-Mendelian. It's a complex trait. So there were There's a history for thousands of years of human beings breeding animals and crops. And lots of the traits that people look at don't behave in a Mendelian way. Whether or not Mendel was the first to get things that act in a dominant recessive one allele way, he was probably a combination of lucky in terms of his choice of traits and organism, and also very, very smart.
And my second question is, since our diet does affect our offspring, are we going to start seeing recommendations for what you do the day before sex or the month before sex, or what you eat the year before sex to have appropriate offspring?
Yeah, so we don't know. So we haven't done experiments where we look at the timing of the treatment. So we don't know whether or not like one day of low protein diet affects kids. To some extent, your question raises the question we think about a lot, which is, what's the bandwidth of sperm? Do you tell your kids that the world was terrible, good, or great? Or do you tell your kids that, I was thirsty when I was 5 years old and there was too much cadmium in the water and this kind of stuff? And so in terms of recommendations, there are two things about recommending things to upcoming parents. One is, in order to recommend something, you have to make some prediction about the future. And the other, so I don't know what the future holds. I mean, water shortages seem like a good bet for most of the world. And the other thing is you need to know how much dads tell their kids. And nobody's going to be good at predicting the future. But the second question, how much do dads tell their kids, is kind of the million-dollar question in my mind in this field.
Thank you.
Thank you.
Really interesting that the low-protein diet is affecting tRNA levels, and I wonder if you've drilled down on whether the low-protein diet can be kind of varied depending on animal sources of protein versus plant sources of protein, and if that has differential effects on.
Yeah, so that's a great question. So what's the sensing mechanism for a low protein? So actually the reason we got into the epididymis is that the dogma about RNAs in sperm is they're the degraded remnants of stuff used during spermatogenesis. So we went and looked at the intact tRNA levels in the testis because maybe it's just the case that on low protein diet, glycine tRNA is up. And those are completely uncorrelated with the tRNA fragment levels in the sperm. So it doesn't seem that the way the sensing mechanism or the regulation of the fragments in sperm works is by just altering the levels of bulk tRNAs. So to some extent, in order for me to think about an experiment to ask that question, requires that we understand whether or not the sensing is done by how long it takes sperm to get through the epididymis, or the charging ratio, charged with amino acids or not, of specific tRNAs, or tRNAs are wildly covalently modified, so we're interested in these modifications listen to metabolites as well. But it's a great question. We can do things like we can starve animals for just alanine or just glycine, and eventually we would like to do stuff like that.
How many generations does it take for these effects to attenuate?
That's a good question. So we've never done so-called F2 experiments. And the reason is just, given that we're interested in mechanisms, our feeling is that as long as we don't understand what happens in one generation, the second generation is going to be lots of knock-on consequences. What I would say is that my read on the literature is that there are a reasonable number of paradigms where you see an effect on grandkids, like the overcalyx cohort. In mammals, it seems like typically 2 generations is about as far as it'll go for most things. It's an interesting, so in worms, when people do RNAi or multiple different experiments like this, it always goes 4 to 5 generations. And I have no idea why it's 4 to 5. And so this question of how long until things decay and how long do you sort of integrate over the environment looks like it'll be different between different organisms, but my gestalt feeling for mammals is that it's going to be 2-ish.
I would just add to that in the human studies, that's as far as we've aged. So it could be 4 or 5. We just aren't that old yet.
Sure, but rodent studies, people have gone to F3s and F4s, and things tend to be dying out around F3s.
That's true, but I would also like to introduce the idea that, as we sort of talked about yesterday, which is different mechanisms, as you've clearly illustrated, so they could produce different effects germ cell So what you're looking at may not last longer than 2 or 3, but it doesn't mean that something else couldn't have been programmed. So that's one of the complex things about risk resilience.
Right. If you looked at 10 million phenotypes, there might be some that go 6 generations.
Ollie, great talk. So just look at the high-fat diet that Lena Clark has reported from Georgetown, by the way. She actually looked at cross multi-generational penetrance. So the high-fat, if it's high-fat corn oil, it actually cannot penetrate through the 3rd generation. But if it is high-fat lard, so the composition of the fat also matters.
Saturated, unsaturated.
Yeah, then it can perhaps penetrate. And then in comparison, for example, an oral contraceptive component that is ethyl estradiol that has a very deep penetrance, it can go through multiple generations. So I find your study extremely interesting, and the kind of priority focusing on the current generation also mechanistically is very, very interesting.
Other questions for our speaker? All right. I think this may be the last one.
This is a layperson's question, and I'm going up to the macro scale. But there are descriptions of raccoons who live in cities being much smarter than their rural cousins because raccoons teach their offspring things that you need to be able to do to survive in cities, which are quite different than in the wild. And so, and it seems to be passed down from one generation to the next. Is that more what, as a social psychology, from a social science point of view, we would look at cultural influences as opposed to epigenetics?
Yeah, so there are, Eva Jablonka describes 4 dimensions of inheritance, which are genetic inheritance, which is your book of instructions, epigenetic inheritance, which is molecular stuff, cultural inheritance, which is raccoons teaching their kids where to eat, or orb spiders teaching their kids the pattern of their web. And then the 2 last ones, there's cultural and linguistic or something. So, like my kids are going to speak English and not Chinese. And they separate between culture, things like the environment building and things that are language-based. Yeah, so all of these are different versions of inheritance, or different kinds of things you can pass on. As molecular biologists, we just study the sort of genetic and epigenetic, but I think cultural inheritance and social inheritance are fascinating in terms of thinking about how we are.
All right. Let's thank our speaker.